no one.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn out cells, but not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we're actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. A little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice, and therefore do PMAT twice. Because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs, called crossing over. Now I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes pop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line, they're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1 where you have two newly formed nuclei and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm, but we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1 though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful as having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction, when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Meiosis sisters, and we remind you to stay curious.
Okay, so that went into more detail than what we need to understand, but a lot of the information is going to be helpful, I think, to you as we kind of move forward. So when we think about meiosis, there's really kind of like some big picture things. The first is meiosis starts with a regular old cell. This is just a regular cell, and it has 2 copies, 2 sets of chromosomes. We're only showing 1 chromosome here, 2 copies of it, so 2 sets of the same chromosome. But these cells would be diploid, or 2N, and the chromosomes are replicated. So each chromosome consists of 2 identical sister chromatids. If we think about where these chromosomes came from, it makes sense to identify one of them as coming from the maternal parent and one of them coming from the paternal parent, but we don't actually know which is which. If we're looking under a microscope, we can't see the difference between this chromosome number 1 and this chromosome number 1. But in our heads, we can say, well, let's call this one the one from mom and this one the one from dad, and then we can kind of keep track of it that way. The first stage of meiosis results in 2 haploid cells. So they only have 1 set of chromosomes, but those chromosomes are still replicated. As the amoeba sisters said, there's something that happens in here that adds a little bit of genetic variation. It's called crossing over. We're not going to focus on that. We can just imagine that this sister chromatid and this sister chromatid are the same, and that this sister chromatid and this sister chromatid are the same. They're not anymore in real cells, but for our purposes, let's just say they are. So this one would still be a chromosome from mom. consisting of 2 sister chromatids. This would be a chromosome from dad, consisting of 2 sister chromatids. During the second part of meiosis, the sister chromatids split apart, and we end up with haploid cells that are not replicated. So we've gone from 1 cell with 2 chromosomes to 4 cells with 1 chromosome. How do we split up 2 chromosomes and get 4 cells? Because each of those chromosomes was replicated, had identical sister chromatids. This cell and this cell have the same chromosome from one of the parents. This cell and this cell will have the same chromosome from the other parent. So we start with 1 cell, 2 replicated chromosomes. We end up with 4 cells. 2 of those are identical, the other 2 are identical. They have non-replicated chromosomes. It's not like earth-shattering, but when you start playing around with it, what you'll realize is the distinction between the sister chromatids and homologous chromosomes, the fact that there's 23 of them, gets pretty crazy pretty quickly. So overall, with meiosis, the real key is that we end up with genetically unique daughter cells, 4 daughter cells, 1, 2, 3, 4 cells from the original diploid parent cell. 2 of them will be the same, the other 2 will be the same, but these will be unique from any other set of 4 cells that are created during meiosis. And that's the main work that you're going to be doing, is focusing on how we can get basically infinite variation. These new cells ultimately become gametes. And in animals, humans in particular, meiosis directly results in the production of sperm and eggs. That's how they're made. In other organisms, not that straightforward, but we're just going to focus on animals. We're going to think about it in terms of humans, because that's what we're familiar with. These gametes are the key to 2 things. 1 is sexual reproduction. 2 is genetics. And that's where we're going with all of this. We're going to try to figure out how it is that, like, everybody looks different. We all have different genes. But if your parents had 2 or 3 or 4 other kids, like, they had the same genetic material to start from. So how is it that we get such different genetic combinations in offspring from the same exact parents? That's what we're going to be focusing on. All right, so first, take 5. I know that was a lot. So take 5, come back at 9:15, and I'll tell you what the next step is.
This one, you got it. Yeah.